Morning to Dr. Hanis. My name is Cindy. Today, I would like to present my presentations entitled The Impact of Bioinformatics in Infectious Disease Research and Vaccine Development. The content of my presentation include introductions to bioinformatics, impact of bioinformatics in infectious disease, impact of bioinformatics in vaccine development, AI and databases in the infectious disease research, challenges, limitations, and future directions, and finally, conclusion. Firstly, let's talk about bioinformatics in the field of infectious diseases and vaccine development. Bioinformatics is the integrations of biology, computer science, and statistical modeling to analyze large scale of biological data such as large genomes and protein sequences. It is essential to use in infectious disease research and vaccine development as pathogens can evolve very quickly, generate a large amount of genomic data sets, and spread globally in a very short period of time. In infectious diseases, bioinformatics enable rapid pathogen identifications, mutation tracking, strain classifications, and outbreak surveillance. While in vaccine development, bioinformatics can accelerate vaccine discovery through epitope prediction, antigen design, and immune response modeling. The use of bioinformatics computational tool enable the transformation of raw laboratory data into clinically meaningful information, supporting faster diagnostic, targeted therapies, and effective vaccine strategies. Examples of applications of bioinformatics include COVID-19 genomic variant tracking, mRNA COVID-19 vaccine design, dengue serotype surveillance, influenza strain predictions, and the detections of TB drug-resistant mutation strain. Next, we will look into the impact of bioinformatics in infectious disease research. Pathogen identification and characterization is one of the most important roles of bioinformatics in infectious disease. Using genome sequencing such as next-generation sequencing, NGS, or whole genome sequencing, WGS, we can rapidly detect and identify the pathogens with high accuracy. Sequence-based comparison also allow early detections of emerging pathogens. Bioinformatic tools also can monitor variant or strain evolutions by tracking the mutations over time. Metagenomic analysis is especially important for identifying unknown organisms or mixed infections that cannot be cultured. Examples include the COVID-19 lineage tracking, such as the Alpha or Delta or Omicron variants that can be identified using genomic approaches. Once the genome are sequenced, bioinformatic tools such as BLAST, Cluster Omega, and COBALT are used to compare sequences, identify conserved or variable regions, and construct phylogenetic trees. These analyses help to detect recombination events, identify zoonotic spillover, and understand how the pathogen evolved. Public databases such as NCBI, EMBL, EPI are important to ensure the global genomic data being shared among the researchers worldwide. Bioinformatics also play a major role in real-time surveillance of infectious diseases. With genomic epidemiologies, we can analyze the pathogen sequence during the outbreaks, thus allowing a faster detection of the pathogen transmission pattern. Phylogenetic tree also can help us to track the outbreak of the origins and to understand how it is being spread across the populations. Genomic surveillance also allow the monitoring of the antimicrobial resistant genes, such as the genes that is responsible for the drug-resistant TB. Bioinformatics also allow the predictions of influenza annual strains to guide the vaccine development. Bioinformatics also support biomarker discovery, which is crucial for diagnostic development. By analyzing large amount of genomic and proteomic dataset, We can identify genes or key proteins that are associated with virulence, immune evasions, and host pathogen interactions. Using in silico prediction pipelines, the researchers can quickly screen and validate potential biomarkers that can contribute in developing various diagnostic tools such as PCR targets, antigen test kits, and other molecular assays. Next, we will talk on the impact of bioinformatics in vaccine development. Bioinformatics is important in modern vaccine design development. By using immunoinformatic tools such as Immune Epitope Database (IEDB), the researcher can predict B cell and T cell epitope directly from the pathogen genomic sequence, which can accelerate the vaccine delivery process. Other immunoinformatic tools such as VacciGem, NetMHC, or NetCTL also can identify antigen peptide, its binding affinity to HLA alleles, and the peptide stability, which all allow a faster and more cost-effective way of vaccine development. Bioinformatics also help to select the conserved antigen regions to avoid rapid mutations which render the vaccine ineffective. It also can perform sequence comparison against human protein genomes to ensure that the selected epitope do not resemble the human protein to reduce the risk of autoimmune reactions. All this approach is to ensure enhanced effectiveness and safety of vaccine. Reverse vaccinology is a type of vaccine development using genomic approach, whereby it involves the analysis of pathogens' genomic sequence to identify the potential vaccine target. The steps start with whole genome sequencing, identification of surface exposed proteins, antigenicity predictions to determine which antigen or protein that stimulate a stronger immune response, and finally experimental validations to validate the selected antigens or proteins where its immunogenicity and safety are being tested. Reverse vaccinology enables faster and more cost-effective vaccine design as it allows thousands of potential antigens to be screened in silico within hours. It has been used in COVID-19 vaccine design by researchers. Next is the structural bioinformatics in vaccine design. In structural bioinformatics, various bioinformatics tools has been employed to determine the protein structure. Protein modeling tools such as AlphaFold, Swiss model is used to provide the 3D model structure of viral protein. The 3D model structure enables the researcher to predict the antigen binding site and to identify neutralizing antibody binding sites to guide the vaccine development. Molecular docking tools such as Autodoc Vina, Pymol also can predict protein ligand interactions, where it helps to determine which ligand or antibodies bind well with the protein. Structural bioinformatics is especially important for mRNA vaccine development. It enables antigen optimization through the identification of the antibody binding sites and the key interaction pockets to enhance the vaccine effectiveness. Bioinformatics also play a central role in mRNA vaccine development. The codon optimization algorithm is designed to help to select the best codon to enhance translation efficiency in cell, ensuring a high antigen expression. The RNA structure prediction tool RNA-fold is used to design stable mRNA molecule to improve protein translation efficacy. All these computational approach allow rapid antigen selections, thus contributing to rapid mRNA vaccine development. With the help of bioinformatics, mRNA COVID-19 vaccine able to develop within 1 year after the outbreak. Next is the CRISPR-based functional genomics. The discovery of CRISPR gene technology had greatly accelerated the vaccine target discovery, whereby this technology had enables a precise editing of viral genomes through the cutting or modifying of the genome sequence to identify the essential virulence genes. This has helped the researchers to identify ideal vaccine targets in the vaccine development. This approach has speed up the discovery and the validation of vaccine targets compared to the traditional method. The CRISPR diagnostic-based tool the Sherlock and Detect also support genomic surveillance, whereby it's able to detect the infectious diseases by binding to the target genome sequences. This bioinformatic tool also allow fast mutation tracking and identification of pathogen variants. Next, we will look into AI and databases in infectious disease research and its clinical impact. Artificial intelligence, AI, and machine learning are currently essential tools in predicting infectious disease outbreak. The machine learning algorithm can analyze pathogen evolutions through the analysis of large genomic and proteomic dataset. For influenza, deep learning model can help to predict antigenic drift, allowing the researcher to predict the next viral strain. Deep learning method also can be used to design multi-epitope vaccines, predicting the most immunogenic peptide with high accuracy. Bioinformatic databases are fundamental in infectious disease research because they help to store and organize all the genomic and proteomic information to be used worldwide. NCBI GenBank provide open access to viral, bacteria, and parasite genomes to support pathogen identifications and evolution study. GSAID is the open access database for influenza and COVID-19 genomic data. Patrick is a bacteria genome database that stores thousands of bacteria genomes informations. UniProt is the key source for high-quality protein information, while IEDP store all the experimental, validated B cell and T cell epitopes that are essential for vaccine design. All these bioinformatic platforms enable global data sharing, faster diagnostic, and more accurate vaccine development. Bioinformatics can give several clinical impact in infectious disease management. These include a faster pathogen identification for a timely initiation of treatment, antimicrobial resistant gene detections to guide the targeted antibiotic therapy, and support biomarker discovery for improved triaging, allowing clinicians to identify high-risk patients earlier. Bioinformatics also enhance outbreak predictions, which enable the public health to be prepared and ready for the outbreak. Overall, it helps to improve patient survival and control the disease. The challenges and limitations faced by the infectious disease bioinformatics include incomplete or low quality of genomic data, limited access to high-performance computing resources, requires skilled bioinformaticians in disease analysis. The computational predictions need experimental validation and also ethical issues in genomic data sharing. In future, infectious disease bioinformatics is moving towards integrating multi-omics, imaging data, and electronic health records for a more holistic understanding of infection. AI will also play a bigger role in predicting outbreak and treatment outcomes. The clinical decision bioinformatics tool will become standardized in hospitals in future. In conclusions, bioinformatics has transformed infectious disease research by enabling a faster pathogen discovery, better diagnostic, and rapid vaccine development. Through strong collaborations between laboratory scientists, researchers, computational experts, and clinicians, bioinformatics can help to turn complex biological data into meaningful clinical insight. This ultimately leads to a better public health strategies and improved patients' outcome. Here is all the reference for my presentations. More reference journals that I cited from. That's all from my presentations. Thank you for listening.